And so in the bacterial quantification by cultured lab, you're counting colonies on a plate, and then you're doing, you account for how many mils you plate on the plate and how many times you diluted it before plating. And then that's how you're able to calculate the actual concentration. So the lab starts out with the same scenario as the last one, where there's some new fungus discovered and you want to see if the compound's antimicrobial. In the last lab, they made a growth curve for you. And in this lab, you have to take samples and count. You plate it and then you count the colonies that form. So a different approach to learning about bacterial growth. And if you were to take enough samples over enough time, you would actually be able to make that growth curve yourself, based on the data from these plates. And it was a little unfortunate that they, at the end, you have 24, 48, 72, and 96. So you had 4 days of data that you counted plates and found the calculations for the concentrations, and then they didn't plot it into the growth curve. So that was kind of a bummer, but you could make it into a growth curve. So anyways, let's go ahead and look at how to do this. So if you have some stock solution and it's concentrated, but you don't know how concentrated, you know that you want to make sure that your plates, you'll have at least one plate that's within the countable range of colonies. So that means colonies that are not touching other colonies that you can easily count. And so typically we would say the countable range is under 100, although some people cap higher, like to 200. And they would just section off the plate into quadrants and just do one quadrant and then multiply by 4. So there's some different strategies, and in this simulation, they are not going to make you count more than 100. You won't even need to count that high. I think 54 might be about the highest that you ended up counting in the lab, so don't panic. And then, so let's continue with this process. So if you have each of these test tubes with, let's say, broth, just pure growth media, and they each have 9 mils, and you take 1 mil from the stock, put it into the first tube, mix it up. Now that tube has a total volume of 10. So let's see if I can actually draw. And it's going to be terrible because I have a mouse and I cannot write with a mouse very well, but bear with me. So in this first dilution, you took 1 mil of your stock and you put it into 9 mils broth. And so that would mean you put 1 mil in, and there's a total volume of 10 mils. And so that's how you get the 1 to 10 ratio here. And if you were to actually divide this, you get this, right? 0.1. So that's the dilution. And when you plate that, the dilution factor that you use will, instead of being 10 to the -1, it's the inverse. It's 10 to the 1, because you want to count, you want to give back the concentration that you took away with your dilution. And then you go, and it's a serial dilution, so you're going to do the same thing over and over again, just making sure to mix it each time and start with a fresh tip. And then you just repeat that a bunch of times. So the next dilution is, it's also a 1 to 10 dilution, but we would say it's 1 to 100 because you already diluted the one before it. So you'll see that in a serial dilution, you go, you increase your dilution by a a decimal place of 10, right? So 0.1, 0.01, 0.001, so on and so forth. And so then the dilution factor that you use when you calculate your concentrations at the end will be, again, the reciprocal. So instead of 10 to the negative 2, it'd be 10 to the 2. And so then we are ready for the next slide. And so when you plate those dilutions, you don't plate the whole 10 mils that would just overflow your Petri plate. So instead, you take a portion, and in this case, and this is the same case in the simulation, you are plating 0.1 mils. Now remember that, oh, I need to select draw again. Okay, remember that 100 microliters is equal to, whoa, I think I went off my mouse pad there, sorry. It's equal to 0.1 mil. So sometimes Labster will tell you that you plated 100 microliters, and sometimes it'll tell you that you plated 0.1 milliliters. And so just remember that that's the same thing. And you need to really pay attention to units whenever you're doing any type of calculation, or else they'll be wrong. And so the equation that you use, if you want to call it an equation, after you've counted the colonies, so we wouldn't count, we wouldn't be able to count any of these. This is complete and total growth. There's not even in colonies. It's just a solid lawn. Here you have tons and tons of colonies, so it's too numerous to count. That's what T and TC stands for, too numerous to count. And even this one, even though you can distinguish colonies a little bit better, is still way, way too many. And so then you get to this plate, and finally you have few enough colonies that you can actually count. And they counted for us in this image, and it's 50. So then we could do the calculation for that. So it's always how many CFUs you count. In this case, it's 50. And CFU is the unit that we use for colony forming units. And then how many mils did we plate? We plated 0.1 mils. So that goes right below, because that's kind of like a dilution in a way. We didn't plate the whole thing, we only plated a portion. And then the dilution factor, DF, is, which dilution is this? This is 1, 2, 3, 4. Okay, so this is 10 to the 4th dilution factor. And you could always put 10 to the 4 in your calculator, right? If you were nervous, if you did that correctly, and you would see, okay, that has 1, 2, 3, 4 zeros, and then you can check, and this here has 1, 2, 3, 4 zeros. So yes, this is indeed 10 to the 4th. And again, it's the dilution factor, so it's not a negative power, it's a positive power, because we want to put back what we diluted. And then when you do this, you end up with a number that is CFUs per mil. So that means in every milliliter of that solution, you have this many cells. And so when you divide by 0.1, you're going to increase by 1 factor of 10. You could also just use your calculator, but I know that instead of being 50, that's going to give me 500. And 10 to the 4th, I don't even need to bother with. I could just keep it. But in scientific notation, we like to do get away with as many numbers as possible. So all these zeros we want to get rid of. We want to put the decimal here. And so I am going to add these 2 zeros places to my exponent. So instead of it being 10 to the 4th, it's going to be 10 to the 6th, like that. Again, you could just put this in your calculator and it will tell you, it'll either give you a 5 with 6 zeros, or it will put it in scientific notation, which would be 5 times 10 to the 6th. And then the unit is here, CFU per mil. Okay, so now if you look at the next slide, I typed it out, what I just had written sloppy on the last slide. So you plated 100 microliters, which is 1 mil. You counted 50, well, they counted for us 50 colonies on the plate. The dilution was a 1 to 10,000. And so that is equal to 10 to the negative 4th. And so then the dilution factor is 10 to the 4th. So another way is saying 1 over the dilution, right? If you want to have a way to put it, the dilution factor is equal to 1 divided by the dilution, and the dilution was 10 to the 4th. Therefore, our dilution factor is 10 to the 4th. You drop that negative. And so when you do all that math, you get 5 times 10 to the 6th power, and that should say CFU per mil. Okay, so now you guys practice the next one. So if you go back on this slide, it says for this one, you got 2 colonies, which technically is not enough to give you accuracy, but let's just do it anyway for the practice. So go ahead and set, you can tell me what to do instead of me telling you. So what do I do? Okay, so the first step, right, is how many colonies. So we said there were 2, and instead of calling them colonies, we call them colony forming units, because the colony, remember, it originated as just a single bacterium cell, and so it's a colony forming unit. And in this experiment, 0.1 mil was plated. So we're going to divide by 0.1 mil. And then we need to know the dilution factor. And the dilution factor here is 1 to 100,000. So that's 1, 2, 3, 4, 5 zeros. So we're going to come back and say 10 to the 5th power. And that's going to give us our CFUs per mil. So 2 becomes 20. And then you could say 20 times 10 to the 5th, or we could say 2.0 times 10 to the 6th. And again, that's CFUs per mil. So whenever you're asked a question like this, you're given either you're given a plate to count, or you're told the amount of colonies, you would need to show this work. You need to show the CFUs, the mils plated, the dilution factor, and then the full answer, CFUs per mil. Now, if you were asked to calculate the actual total bacteria in the original sample, you would need to know the volume of the original sample and multiply by the volume. But we didn't do that in the lab, so I didn't ask you to do that for the homework either. Now, for this, I'm asking you to decide which plates do you count and then you should count them and then do the calculations. So obviously this one is not even forming colonies, so it's too numerous to count. Same here, right? And now I want you to use your judgment to tell me about this plate, and this plate. Okay. So I'm not going to give you the answer here. I can give it to you next week after it's due, but I asked, giving you the opportunity to get 1 bonus point by doing this on the homework. Okay. So I'm not going to give you the answer yet. Now that's pretty much it for the bacterial quantification by culture. It sounds hard because they use quantification, but it's actually probably a bit easier than some of the math we've already been doing so far. So that's it. Now for the growth curves, kind of went a little backwards, but for the growth curves, here we go. So first you want to know that bacteria growth is by binary fission. So 1 cell, in fact, let me go ahead and draw again. So 1 cell divides and becomes 2, right? And then both of those cells will divide, and so you'll be left with 4, and both of those divide, and so you'll be left with 8. Sorry, that's sloppy. Not that that's going to make it much better, but it's a little better. And then each of those divide, and so then you're going to be left with 16, and so on and so forth. But you know, you would never, like in a solution when you're growing bacteria, you would never have just 1 cell. You would start with a much larger population. The only time you would really get down to 1 cell is when you've been doing the streak for isolation method and you've been trying to dilute it down to just individual cells to grow colonies. And that's why plates, sometimes you can incubate for just 24 hours, but sometimes you need to incubate for 48 or even longer, because you have diluted down to just 1 cell, and you need to give enough time for enough divisions to happen, because you need to get in the, you know, hundreds of thousands, if not millions of cells, before you can get a colony that you could see with the naked eye. All right, moving on. So you, in this lab, you're dealing with broths, growth media, and you want to make growth curves to see how quickly do they grow, and what are the phases of growth for that sample. And so some factors of bacterial growth, which I'm sure you're familiar with from lecture, but let's review. So temperature is the first and biggest concern when you're growing a culture. You should always make sure that you're incubating at the correct temperature. When you're making your media, you should make sure that the media is at the correct pH. Usually this will be around neutral, and usually the temperature would be around body temperature, which is 37 degrees Celsius. But each type of bacteria will have its own optimal temperature and pH. Of course, water availability is not an issue in the lab because you're already giving it media, which is mostly water. And of course, the nutrients and the composition, you would make sure you're using the correct nutrients, and usually that's also not an issue, but sometimes you do have to give special additives depending on what you're doing. So those are the main factors. Of course, there's more, but these are the main factors that you would need to be thinking about. And then, of course, you grow it and you make your growth curve. So to make a growth curve, you would need to have samples taken frequently over usually days, so that you can get this growth curve. The more data points you have, the better the curve will be. And from that, you can calculate the generation time how long does it take for each of these divisions to occur? And we take these during the rapid growth phase called the exponential growth or logarithmic growth phase. And then we can calculate the growth rate constant. And again, that would be for the exponential growth because it changes. And in fact, let's go ahead and look at the next slide so you can see. So bacterial growth, it looks like the bottom of this got cut off. Wonder if I can fix that real fast. I guess my screenshot didn't catch the bottom, but on the bottom would just be time. And on this side is the log number of bacteria. So instead of using the whole number, which is in the millions, you take the log of each of those and you get numbers that are easy to work with. Otherwise, you wouldn't be able to really make the plot. And when you do, you'll see that usually the first few hours, sometimes longer, depending on conditions and bacteria. But the bacteria, when you first put them into growth media, they need to sense the environment you put them in. What kind of nutrients are available? How nice is this environment? And once they sense that there's plenty of nutrition and what kind of nutrition is available, then they can start synthesizing all the enzymes that they need to utilize that and also start multiplying their DNA, getting ready for everything they need to do to get ready for rapid division. And so then once that's all ready, and it begins, then they'll start just dividing very quickly at the same constant rate. And because it's at the same constant rate and each cell divides, you get a doubling effect. And so that makes it exponential growth until they start to run out of nutrients and also their waste products start to accumulate. So they get these cues, well, we're starting to run a little low on food. We're starting to change the pH a bit. And so they slow down and they will just divide slowly at a rate that's equal to the death rate. And so you get the stationary phase is the amount of new, cell division is going to be equal to the amount of cells that die. And then eventually they eat all of their food, or they've created so much waste that it's not inhabitable for them anymore, and then they die. And of course, this is not instantaneous, but it is a slow process because the weaker cells will die first. The stronger cells will last a little longer, but eventually there's no food or the pH is too different and they die. Okay, so that's the death phase. So that's really the biggest part of the lab. And then it's just using the growth curve to do your calculations. Okay. And of course, the overall idea of the lab is using the growth curve, using these calculations so that you can determine if the fungal compound is an antimicrobial compound or not. They also give you a little history of antibiotics. I decided that's not really the purpose of the lab. We will have another lab that's devoted to that. So I'm not going to ask you about that for this lab for this week. Okay, so I just went over this and I included the descriptions. So I talked about it, I described it a bit here, but I decided it's always best for you to have it in writing with all the information. So I included these here for you, for your reference, but I'm not going to read these to you right now. Okay, and so then you just want to see some of the calculations that they ask you to do. So in this bacterial growth curve lab, they do serial dilutions and colony counting for you. They tell you that that's a method you can use, but then you don't actually do it until the next lab, which is completely devoted to that. So we don't need to talk about this because we already did, because we did the other one first today. Okay, so generation time, as I already mentioned, is how long it takes for the cells to divide in half. And so you get a doubling of the population. You would need to know how many cells you started with. So capital N is the population size. And when you are working with a population that can change, you would need to denote the time point that you're referring to. So we actually would say N naught is how many you start with. In this picture, you're starting with 1, but in most situations, you're starting with hundreds, thousands of cells, okay? But the first time point, you could take a sample, plate it, count plates, and determine how much you actually started with. In this lab, I think they just tell you. And then N1 is some time point after N naught, and N2 is some time point after N1. And just putting the T, the little T, just means some time, whatever time you decide to put in there. And so this would be the population size at some time, and that time is not determined specifically, but just saying like at some point. Okay, so the equation then for generation time is that in T, whatever you're interested in, is going to be equal to, so your population size at some time period, let's say 2 hours, is going to be equal to your population size at time 0 multiplied by 2 to the nth power. And n is, the little n is going to be your, I think I put it here, number of generations. Okay, so here I broke it down for you, even though it is also here. So NT equals 2 to the nth power times N naught. And N naught is your starting population size. And 2 to the nth power would be a doubling every generation for however many generations you decide. And if you have that, then you can find your total population. Now, if you wanted to know, I'll come back and talk about this in a minute, but if you wanted to know what is the generation time, then you would need to look at the amount of time you would divide the amount of time by the amount of generations. Okay, so here it says by counting the population at N naught and NT, you've calculated the number of generations which occurred in the interval. So you found that little n, and then you can use the little n that you found to determine the length in one generation, or here it says by dividing the time of the interval t by the number of generations, little n, you will get the generation time for one generation. And that's the capital G. So one generation is the total amount of time divided by the total amount of generations in a sample. And I know this is kind of hard when you look at it, but then when you get to use it in practice, it'll make a little bit more sense. And lastly, the last calculation is growth rate constant, which is K is equal to N divided by T. And so that's taking what you did in this previous equation and flipping it upside down. So I don't know if that answered the questions you were asking. One more thing I want to talk about before I open it back up for questions. I mentioned how we don't plot using actual numbers because they double every time. Your numbers get so huge that you can't even fit it on your plot. Like, first of all, from 24 to 72 hours looks like nothing happened, even though a huge amount of growth happened. And then it looks like they really took off, even though it's actually been the same rate that whole time. Um, and then after this time point, you can't even fit it on this plot anymore. It's just way off the chart. And in fact, they gave you a cute example that if you were to try to plot the next data point, it would be 500 meters in the sky. Like, that's how, this exponential growth is, so it gets such huge numbers that there's no way to physically plot that number unless you were to change your scale so much that it's not even useful. You can't gain any knowledge by looking at it. So to solve that problem, we use the log scale on the y-axis. And so now you have nice numbers again, like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And so using those same numbers that we saw over here, we plot it using the log scale, and now we get this nice little like straight line or a slight curve, right? And you still keep your incubation time in hours. That did not change, but the only thing that changed was the y-axis for log. Okay, so hopefully that helped you understand why we use the log scale. With the math, you really just have to practice. You can read it as many times as you want, but until you really go through the examples and practice it, it might not make sense. And then this is the post-lab questions I asked, and I put here an arrow to point out that I did make the attachment. So to answer number 4, you need to use this attachment here. And that is it. So I'm going to